Tell me when. Good? Are we live? Okay. All right. Hello. It's a pleasure to be here with all of you today and with our online audience as well. My name is Brian Rood. I'm the Medical Director of the Brain Tumor Institute at Children's National Hospital. And it's my pleasure to moderate today's session of our keynote speakers here today. We have 3 distinguished speakers. I'll introduce each in turn. For those of you who are in our online community, please enter your questions into the chat, and they will be sort of collated. We are, however, going to hold questions till the end of the session, and we will sort of, parcel those out as appropriate. But if you enter them sort of real time while they're in your mind, that's fine. We'll grab them and make sure they don't get lost. So I have the pleasure of kicking this off by introducing Haneen Shalabi, who did her training at the Chicago College of Osteopathic Medicine, and then, of course, where I met her at Children's National Hospital, where she was a Hematology Oncology Fellow of some renown. She then went on to the National Cancer Institute, where she's been working in Nurali Shah's lab on better ways to target chemotherapy-resistant pediatric leukemias, particularly using novel immunotherapeutic approaches. Today she's going to describe her current work for us on a clinical trial with a novel CAR T-cell construct, work that has been supported by the CCF. Thank you, Haneen.
Thank you. I'd like to thank the CCF for giving me the opportunity to speak today. It really is great to be back. It's been a few years since I've been here, so I look forward to presenting our updated work to you all. Today's talk will be focused on chimeric antigen receptor T-cell therapy in pediatric acute lymphoblastic leukemia. And this is my NIH slide. I have no disclosures to report, unfortunately. As you all know, acute lymphoblastic leukemia is the most common childhood malignancy, and there's approximately 3,500 new cases per year in the United States. Upwards of 85% of those with standard risk ALL will be cured, and this is in large part due to the combinatorial chemotherapy strategies and the de-intensified and intensified treatment approaches, utilizing different chemotherapies with non-competing mechanisms of action, and additionally implementation of CNS prophylaxis over the last several decades. This in turn has allowed for improved overall survival in these patients. However, a significant problem still remains in the relapse refractory ALL setting, and this presents an opportunity for novel therapeutic discoveries. Chimeric antigen receptor T-cell therapy has really revolutionized the treatment of relapse refractory B-cell ALL in pediatric patients, and this is depicted on the slide. A CAR is a genetically modified T-cell utilizing the best of the B-cell, which is the antigen recognition domain, in combination with the intracellular functionality of a T-cell to create this immunotherapy, whereby essentially your body is able to target your tumor cells or antigens that are expressed on your tumor cells in a more targeted fashion. The premise of CAR is essentially to modify your cells so that they can recognize and destroy cancer cells. As relapse remains a problem, and the mission of the Pediatric Oncology branch at the NCI is to find novel ways to treat and address current limitations in cancer therapies, we embarked on a journey to determine how we could better utilize immunotherapy in pediatric patients. So with the premise known that foundation of ALL has been improved and treatment strategies were improved with combinatorial chemotherapies and implementation of CNS prophylaxis, we thought, well, why can't immunotherapy be any different? With CD19 and CD22 CAR T-cell single antigen targets, remission rates are very high within the first month of therapy. However, there is a significant problem with antigen escape. And so we thought we could potentially do combinatorial strategies with CAR T-cells, and there are several ways that this could be done, and these are depicted on this screen. So co-transduction is basically when you take 2 vectors imported into a mixed bag of cells, and then you can come out with a mixed population. Some are single antigen positive, some actually co-transduced to have double antigens on the surface, and then of course some don't get transduced at all. There's a co-infusion mechanism whereby you do 2 separate CAR products, and then you would co-infuse the patients with each individual CAR product. And then there are additional ways to achieve combinatorial strategies using a bicistronic approach, which is utilizing 2 vectors within 1 T cell that gets split when it gets transduced, or using a bivalent approach, which is depicted on the screen, which is essentially a tandem CAR. And that is the trial that I'm going to talk about today. And so with the preclinical work at the NCI and garnering all of the experience from our CD19 and CD22 CAR T-cell trials, we embarked on a journey to create a bivalent CD19, CD22 targeted CAR T-cell product. And preclinical data demonstrated efficacy and in vivo activity and significant CAR expansion in the xenograft models. And so the novel humanized bispecific CAR construct was developed using the FMC63 CD19 scFv along with the M971 scFv. And that's where we initiated our bispecific CD19 CD22 CAR trial. So to date, we have treated 19 patients, and this is a 3+3 dose escalation study. The phase 1 study has now sort of moved along into the dose expansion phase, so 3 dose levels have currently been explored. Our subjects represent a heavily immunotherapy pretreated population. 70% of our patients have received some type of immunotherapy before coming to our trial, 50% of which have received stem cell transplants. 50% of patients treated to date have either an M2 marrow, so greater than 5% blasts in their bone marrow, and or extramedullary disease. So a significant proportion of patients treated have high burden disease. Additionally, we, as you can see in the waterfall plot, have demonstrated early clinical activity with this CAR construct, and really it was a dose-dependent manner by which we had remission rates. And so of the 19 patients treated at dose levels 2 and greater, we've had 10 of 19 patients achieve MRD negative complete remissions. Especially in those that had high burden disease, we saw significant CAR expansion. We additionally noted that toxicity was reversible in all patients and noted that 3 patients of the 19 had greater than or equal to grade 3 CRS, so requiring ICU level care, and 1 patient had neurotoxicity that was reversible. We did note that patients with higher burden disease did have a predilection to develop more significant cytokine release, and this was consistent with previous studies that have been reported. Limitations to this experience to date include the fact that our CAR T cells in all responders have expanded, however, persistence is lacking. So patients have had MRD negative responses, but then within a median of 45 days, we are not able to detect these CAR T cells. Additionally, in those with extramedullary disease, we've seen complete resolution of bone marrow involvement. However, extramedullary disease has persisted, and so trafficking to sites of extramedullary disease is still an active area of investigation. So there's several ongoing correlative studies that are needed and that are being currently performed to optimize these treatment strategies. So as I've noted, we have limited CAR persistence, and with that, out of the 10 patients who are in remission, we've had 4 patients relapse, 3 of whom had antigen-positive disease. And so there's a significant amount of work that needs to be done in order to understand the leukemia biology, looking at the immunophenotype of relapsed disease. Additionally, understanding the patient cytokine profile, so those are currently underway, looking at CAR T-cell trafficking persistence, and expansion, especially in the blood, bone marrow, and CSF, and also trying to note why extramedullary disease is so difficult to target. So potentially introducing something like a checkpoint inhibitor into CAR T-cell therapy to try and overcome that barrier. We're also looking at immunogenicity studies. Although the CAR is a fully humanized scFv, there are specific areas of the CAR construct that could potentially have been mutated, and so we would like to evaluate whether patients have immune-mediated rejection or not. So these studies are also ongoing. Additionally, in an effort to evaluate responders versus non-responders, we are looking at patients who have responses and evaluating and analyzing their CAR T-cell product characteristics. And then finally, we have an upcoming amendment to enroll CNS-only disease patients to further look at safety and the toxicity and efficacy profile in this patient population. So I hope that you guys just got a small glimpse of what we're doing at the NCI with the help of our friends here at CCF. And so it takes a village to run CAR T-cell trials, and I would not be here without my village, which is the POB. I am forever grateful for their opportunity to really move into the clinical world and really translate bench to bedside and back. It's such a fantastic place to work because you really, in real time, have patient cases that really cause you to think, drive you back to the bench, collaborate with colleagues, and then in turn make clinical changes. And so with that, I will stop and pass the baton along, I think. Thank you.
Thank you, Haneen. That was wonderful. Look forward to the question and answer period for your talk. I next would like to introduce Dr. Jeffrey Toretsky, I think who everyone here knows. Jeff has really been a longtime friend and mentor, certainly to me and to many, many people in this audience and in this region. It's sort of one of his, I think, things that really drives him is the introduction or the excitement around science and around bringing more people, young people into the fold. He's been a tireless quester also for better treatments for children with sarcomas, most especially Ewing sarcoma, which has really been quite a challenge, certainly throughout my career and certainly prior to that as well. After his training at the University of Minnesota, he was at the Medical College of Virginia and then after that the National Cancer Institute. And he's really based the majority of his career at Georgetown University, where he's currently the chief of the Division of Pediatric Hematology Oncology and leads the molecular oncology program for the Lombardi Comprehensive Cancer Center. He's the chair of the Scientific Advisory Board for CCF and has made a tremendous contribution to the success of CCF, and I think was also the inventor of this symposium, which is now in its 5th year, and was really a wonderful idea that, sort of I think when it first happened, a lot of us went like, geez, why haven't we been doing this for a long time? It makes a lot of sense. Today he's going to tell us about his support, about how the support from CCF has really helped him to pioneer a therapy targeting a historically difficult protein in Ewing sarcoma.
Well, thank you, Brian. And we were sitting down here. Dr. Civin turned to me and said, "Should we be standing up during this?" I'm like, it's hard to give a talk sitting down. I realize that I want to jump up, so if I just jump up with enthusiasm, it's just hard. Okay. So yeah, this is a 20-year quest, and it's really interesting because this could not have happened without CCF, and many of you knew Shirley, but fewer and fewer over time. She was a phenomenon, is the best way I can describe her, and she loved raising money for childhood cancer, and she made it possible for me and others to get a start. And you'll hear about that some more from Dr. Civin as well. And she would have loved the work, Dr. Shalabi, that you're doing. And unfortunately, she had died before that, but she would have loved that. So my disclosure slide. Yeah, fortunately, in this case, we do have some disclosures. Small molecules are patented. I should update it because they're less and less are pending, but every now and then I get a postcard from like Korea or Japan and letters that I can't read, and it says my new patent, they've issued a patent, so it's kind of fun. It's been licensed to Nocturnal Therapeutics, and I'm a founder of this company, and we can talk more about that. So Ewing sarcoma, as Dr. Rood alluded to, is a really awful disease, which fortunately for patients with localized disease in this purple curve, we've made a small improvement over the last 30 years, but really not enough. And for patients with metastatic disease, the outcome is still dismal. And in fact, just this week, I'd learned that a patient of ours that was actually a patient between King's Daughters Children's and myself sadly died of his Ewing sarcoma. And so we have to do better. And the father wrote me and said, you know, one of his son's wishes was to keep doing it, to keep working toward this. So because this is sort of a historical retrospective, I went back and I found about 40 slides from talks that I'd given 20 years ago, and then I got rid of all but one of them. And but I want to point out that in 2004 at a symposium by the American Society of Peds Hem-Onc, I was up talking about potentially targeting this thing that I'm going to talk about today, which is the fusion protein EWS fly. And at the time, there were some other approaches as well that I don't have time to go into, but focusing down on finding the right target, which had kind of already been given to us, but then how to do it, and the chance to make mistakes and to learn from those mistakes and think about how to go forward was a gift that CCF gave me because it funded trials long before the NIH was going to give us funding for that. So our goal for Ewing sarcoma was to improve the therapeutic index. And by that, as we learn about new drugs, it's really important to keep in mind what this therapeutic index is, because if you don't improve the therapeutic index, the new drugs aren't going to be better than the old drugs. Basically, it means that the amount of drug, or the milligrams, that it takes to kill the patient is in a ratio with the amount that it takes to kill the tumor. So here I give an example where if it takes 100 milligrams to kill the patient and 1 milligram to kill the tumor, that's good. That's a good therapeutic index. But a bad therapeutic index is more like 1.5 to 3, which is what our current drugs are. So we have to do better. So where are we going to do that? Well, it turns out, oh, it did change the fonts. How about that? Oh, that's okay. I'd been warned that the fonts might look funny. So it turns out that in Ewing sarcoma, there is this chromosomal translocation. And this is what I meant by it was sort of given to me. It had been discovered in the 80s. And then in 1992, right at the time when I was starting my work as a fellow, the actual genes on either side of that chromosomal breakpoint, where you can see here on the slide, 11 and 22 break and trade places, you can see that the two genes, EWS and FLY1, each break at that point, and they come together and make a totally new protein, which we call EWS fly. And literally for the last 20 years, we've been studying how EWS fly works. And one of the things we've learned is that EWS fly does not just do one thing. It turns out that it does a lot of things. And as we study how it works and think about how to turn it off, we have to keep all of those things in mind. It's involved in transcription, making new RNA. It's involved in splicing, taking that new RNA that's made and putting it together in the right way so that when it goes out to the cytoplasm and becomes a protein, it actually becomes the correct protein. And then also, we now know from studies that have been coming out is that it's also involved in DNA repair. And this is an area that we're starting to get into. So in order to turn off EWS fly, in order to inhibit it, when we started thinking about this 20 years ago, we heard words like it was undruggable, it wasn't like BCR-ABL where you could put a kinase inhibitor in a pocket. And so we really had to use our imagination of how we could turn this thing off. And the way we did it, before I tell you, first a little thought exercise. Use your imagination. It's one of the things that we just don't do enough of after kindergarten in our lives. And I really think it's important. So start by imagining a color you've never seen before. And that's what we had to do with turning off EWS fly, okay? So I don't know if you saw one of those colors emerge, but that's where we had to start from. So what did we try to do? In this slide, this is a simplistic version of what we tried to do. So here's EWS fly. And we knew that it bound to DNA, so I've shown you that. And there were these other boxes, and it was like, well, we know it's also going to touch other proteins. And the question is, what are the proteins that it's going to touch? And the first work that CCF funded for us was finding out that RNA helicase A was one of those proteins. Okay, well, that's great, but now the question is, how do we turn that knowledge into a drug? And we thought about if we could make a drug stick to EWS fly, but not stick to RHA, which was a normal protein in the cells, we could, in theory, kill Ewing sarcoma. okay? So it would look kind of like this. So a new drug would actually stick to EWS fly and block other proteins that EWS fly needs to do all of its dirty work, and that would allow us to kill the tumor. So in another cartoon form, it looks like this. Here's EWS fly, here's RNA helicase A, and that little green oval is our imaginary drug. And the question is, how do we find that drug? Well, it turns out what we did is we used a technology called surface plasmon resonance. And what that basically means is we put EWS fly on the surface, and then we run chemicals one by one over the surface. So now you are inside the machine that we use to discover the drug that will stick to EWS fly. And that was the original goal. Just could we find a drug that sticks? And if we could find a drug that stuck to EWS fly, and you can see here, it bends the light. Whenever something sticks on the surface, it bends the light. And along comes our little drug, and we ran drugs one by one over EWS fly, and eventually we found some that stuck. And when we turned off the drug would go away, and you can see the light bends back, and we were looking for the ideal curve like this for a small molecule, a chemical, as you will, that would become then a drug. What we identified from what we call a library of chemicals. Now we're getting, a library of chemicals is nothing more than a bunch of little dishes, and each dish has about 100 chemicals in little tiny wells in the bottom, and one by one we would put these dishes into our machine, and we found one, the NSC compound here, that stuck to EWS fly, and then we did some chemical modification, and we found one that was a little better, which was YK4279. And the reason it's called that is it was synthesized by Yali Kong, YK, and her fourth notebook since coming to Georgetown on page 279. Now, one of the problems we had is that since this compound stuck to EWS fly empirically, we didn't know where it was sticking. And this has led us to a whole adventure in the last 12 to 15 years, which I won't have time to go into, but we had the benefit of knowing that one of the carbon atoms in this molecule was chiral. What chiral means is that it means that there's a left-handed and right-handed version. So that arrow is coming from one of those carbons where one of the groups goes into the screen and one of the groups comes out of the screen, and we call those enantiomers. And it turns out that in a whole series of studies, only one enantiomer, the left-handed one, the S enantiomer, worked. And I'm showing you a growth assay, I'm showing you a binding assay, a transcription assay, and a helicase assay. They're all annotated at the bottom, and I don't have time to go into the details. But the key take-home point is that one enantiomer, chemically identical to its opposite, works and one doesn't. And that allowed us to at least declare that there's some specificity to how this molecule is actually working. So then we went on and we found out that there were a lot of troubles doing this study in mice, so we went to rats. We created the first rat model of Ewing sarcoma, and what this showed is that after a continuous infusion over a series of days, we could literally cause the CD99 cells in the leg of the rat to completely be gone. So we literally cured a 2-centimeter tumor, about so big, from a rat to be completely gone in 5 out of 6 rats. So that led us into the clinic. So it turns out in 2009, we reported the YK4279 in Nature Medicine, and immediately I started getting emails from family members and patients with Ewing sarcoma. When does this become a drug? So, unable to license this idea to a company, I decided I would try to start my own. So I started my first company in 2010. We called it Targeting Disordered Proteins 1. It failed. Then there was Targeting Disordered Proteins 2, and it failed. And finally, in 2013, going into 2014, I met a serial entrepreneur named Scott Glenn. And Scott was somebody who had a history of starting companies. So he took our technology, started a company around it, and then they did some extra chemistry, and they created an analog of YK4279 called TK216. And in January of 2016, the FDA granted the investigational new drug application, and we were off to the races. There's been a phase 1 clinical trial, which is now concluded, and we're now into a phase 2. And let me show you a little bit about what this looks like. I'm going to skip the phase 1 trial escalation for now, and I'm just going to show you one example of a patient. So this is a patient who presented with metastatic Ewing sarcoma when he first walked in the door. So his survival already was going to be poor. He had 4 relapses. He got 3 different salvage regimens over 2 years, okay? In February to March of 2019, 19, he enrolled on the trial, and after 2 cycles of just TK216, his chest CT scan, shown here, showed 3 out of 4 nodules completely disappeared. A 4th nodule was left as a small residual, and in August of that year, he had it resected. Okay? Since that time, he's remained on TK216. It's over 2 years, and he's still in remission, which for a Ewing sarcoma patient is really quite remarkable. And we've got other patients that have had, another patient has over a year response, and others are coming along. So one of our challenges now is to understand the difference between the responders from the non-responders. And that's one of our areas of continuing research. So I just wanted to share this slide with you because CCF was so responsible for getting this going. I wanted to give people a sense of the benefit, and not just of the drug, of all the benefits that have come out of this. And so this project started with an idea, not quite on a napkin, but about 20 years ago. And for the first 8 or so years, CCF was providing seed funding, over $1 million. And then finally, after we got that small molecule lead, we started getting NIH funding. So that came in over the next few years at about $8 million, including some Recovery Act funds. And then I also was fortunate to get a Burroughs Wellcome Physician Scientist Award, which also helped the research move forward. And there's some patents, and there's a startup company, and we have a drug in a clinical trial, and we have at least two patients in complete remission now, and lots of mentoring of students and new faculty along the way. So all of that together is something that, my gratitude for CCF is significant. It didn't happen alone. Acute has been an incredible partner in this journey ever since he joined my lab in 2000. We've had a tremendous amount of fantastic students through the lab. I'll just point out Stefan Zellner was a fellow with me. He's now in Essen, Germany with Ute Dirksen as a professor running a lab himself. So it's a really, really great opportunity that I was very fortunate to give to him. And of course, CCF has seen these slides over the years. So thank you very much for all that you've done. And with that, I will pass the baton to my esteemed colleague who preceded me and the CCF, Dr. Civin.
So before you do that, I have the distinct honor of welcoming Dr. Civin and introducing him. Dr. Civin has really been sort of a mainstay of the Washington, Baltimore sort of regional pediatric oncology community for many years now, and for many of us has really shown the way towards a successful pediatric hemonc career, and the fact that one does not need to work in breast tumors and prostate and all of these adult cancers. One can be a successful scientist in pediatric oncology, and that's really, I think, shown the way for lots of us. Dr. Civin is the discoverer of the CD34 stem cell marker. I think that's something all of us have learned tons about over the past number of years, and is also a pioneer in the development of antibody-based therapies for children with leukemia. And this is particularly an area that's just yielded a host of now proven therapies for many different types of pediatric cancers. And we're seeing it in the clinic all the time now. After his training at Harvard and Boston Children's and then the NIH, he began his career at Johns Hopkins, where he worked for quite a number of years and is now a professor of pediatrics and physiology at the University of Maryland. He's currently the Associate Dean for Research and the Director of the Center for Stem Cell Biology and Regenerative Medicine. Dr. Civin was there at the beginning of the CCF, and he's now going to tell us about how CCF support has helped launch his seminal work.
Thank you for that glorious invitation, Brian, and glorious introduction. Excuse me. So about this talk and its title that you see there, stolen from Dr. Seuss with attribution. Jeff asked me to give this talk pre-pandemic when this was going to be scheduled. And he said, I think, that, well, what you should do is in this panel, in this talk, you should say how the Children's Cancer Foundation helped you and everything, and maybe take a little bit of a victory lap if you want to. So I'm foolishly going to try to give a talk with this title here. It's a little different from most of the talks that you've heard, but a little bit like Jeff's. And I just want to thank you all this morning for a lovely opportunity for me truly to thank the CCF for support of research that's still impactful today, 4 decades later. Really, if you think about the CCF's history of near 40 years, and you think about a generation of physician scientists, a generation is about 3 to 5 years or something like that. So that's 10 generations of pediatric oncology support here in Baltimore, Washington. That's disseminated for the nation. So thank you, CCF. And I see in the back a friend and one of the most recent supporters, generous supporters of Children's Cancer Foundation, Greg Keller. And I want to thank Greg, too. So back to about 1980, and I don't know the exact year, but in those prehistoric times when most of you were not born yet, I was a young pediatric oncologist starting my lab at a brand new cancer center called the Johns Hopkins Oncology Center that was started, I think, in 1976. And I was, I came, as Brian said, from Boston. And part of the way that pediatric oncology flourished in Boston was with the support of the Jimmy Fund. How else would they get support when pediatric cancers are not so common? And so we said, we need a Jimmy Fund here in this region. And we got together, again, as I remember it, with Shirley Howard and her husband, Bill Howard, and 3 pediatric oncology families, the Mitchell family, Ginny Mitchell, Jerry Kelly, and Bernie Shanahan and their families. And we had a meeting, and there were maybe 6 of us there, or 8 of us there, and we said, let's start our own Jimmy Fund-like organization down here. I also think that I was the first CCF grantee, with no guarantees, grantee, but in contrast to Jeff, I think I got $15,000, or maybe it was $17,500 or something like that.
Accounting for inflation.
Yeah, it was so back, it was in, clamshells or something like that rather than bills. So at that time, as Brian said, CCF supported my absolutely crazy idea to produce monoclonal antibodies against stem cells. At that time, the leading lights in the field said you make monoclonal antibodies against the mature cells, and you just pull them out of the way, and there will be the stem cell. Well, that didn't work. But so we made the hypothesis, and it was pure hypothesis, that there would be some stem cell antigen that was expressed in large amounts on leukemia cells, so we could use them as immunogens since we couldn't purify the stem cell, and that we could inject them into mice, which was the technology at the time, now evolved into CD22 and CD19 antibodies that we made with similar techniques. And that these antibodies on leukemia cells would be shared by normal stem and progenitor cells. So we immunized a lot of mice and screened for thousands of leukemia lines. And if there was a lesson from this, screened against lots of leukemia lines, screened thousands of hybridoma cell lines, was that we had to throw out 99.99% of these antibodies. Some we licensed to companies for other purposes, but we made the antibodies against B cells, myeloid cells, NK cells, and we said, no, we don't want these. What we really want to focus on was something that doesn't bind to any, ideally, cells in blood. And I remember when we knew we had this, we said we've got to be able to commercialize this. And we sent the monoclonal antibody to Becton Dickinson, and they tested it on marrow, and they said, it doesn't bind to anything. And we said, yeah, that's right. It only binds to a tiny percent, and we can purify the cells, and they look like this. And they said, oh, okay. And they went back, and they said, and they licensed it. And so, I wouldn't be telling this story or here today if the idea didn't work. So the hypothesis was validated, and the antibody became a reagent and the basis for a technique for identifying or purifying stem cells. So obviously, you all know that that purified CD34 positive cells have become a workhorse, and there are thousands and thousands of papers that study CD34 cells or use CD34 as a reagent. It also stains endothelial cells. Judah Folkman once told me it was his best antibody for identifying endothelial cells. I said, okay, a side reaction. It wasn't as specific as it should be, as I'd hoped it would be. But this led to an interesting consequence that when citation indices became utilized, I had the problem that most of my work was no longer cited. It was just part of the wallpaper in kind of taupe or whatever color that is. But we could use these antibodies to really trace the developmental processes in the marrow, which are much like the developmental processes in every other tissue, in that you start with a very early cell and you progressively mature over time. This doesn't seem to point, but in these pictures you can see cells that start out, and there's not one cell. There's a cluster of cells with different levels of expression of the two antigens that are being used here. Now we use multicolor, polycolor flow cytometry and single cell sequencing to do the same thing as these two antibodies here in the picture. But even normal hematopoiesis is somewhat heterogeneous. I sometimes say that it doesn't really matter if I put on my shirt first or my pants first, just so that when I came here, I had both on at the point that I'm the mature cell, hopefully. So we could use CD34 as the anchor to identify the earliest cells in the population, and then we could use other monoclonal antibodies or nowadays different reagents to trace erythropoiesis here, or to trace B lymphopoiesis here, or to trace myelopoiesis or development of eosinophils or whatever lineage. But so at that point we said, very nice, we have a very expensive right stain, but the neat thing here was we could isolate every cell in this evolutionary developmental pathway, and we could study those cells, and we could study their bad twins and their aberrant twins, the leukemia cells that arose from this compartment. But then somebody said to me, well, CD34, that was supposed to be a stem cell antibody. And really 99% of the cells that you have are progenitors, which are far beyond the stem cell change. And I said, yeah, okay. So let's find the stem cell, and we and many other people, Irv Weissman, made collections of monoclonal antibodies where CD34 again served as the anchor, and several other antibodies served to discriminate here as CD38, the more mature progenitors from the stem cells in the compartment. And nowadays we can do a fairly good job of that, but now we still can't isolate perfect stem cells, and perhaps that's biological. Perhaps we're thinking today that the differences between stem and progenitor cells aren't as stark as we thought they once were. So I mentioned commercialization, as Jeff did. That's the, we're all physician scientists here, and I often say to people that if this is my scientist hand, and I've got, say, a monoclonal antibody, or it could be a drug, I can show that it works, I can develop this, but I can't put it into my other hand, my physician hand, and try to see if it works, make it work in a patient. I need industry in between that. The university that I work at isn't going to make pills and do all the non-trivial chemistry and medicinal chemistry and formulation that has to be done, not just the marketing. That was the rationale for patenting CD34, following in the shoes of others who had patented monoclonal antibodies against T and B cells. And this was the patent, and it caused me for years, as long as the patent ran, unfortunately it's now expired, to have to disclose a conflict of interest of this. I no longer have to disclose that you should take an extra grain of salt. Take whatever I say with an extra grain of salt, because it might be supporting my kids' education too. So when we licensed this eventually to Becton Dickinson, the general counsel of Becton Dickinson, who made the decision to license this, said to me, well, congratulations, Curt. If this antibody does what you think it will do, you're going to be sued and in court. And I said, what? I mean, we so clearly preceded anybody else making a CD34 antibody, and the next paper that came along actually cited us and said we did it the same way to make a different monoclonal antibody against the same antigen. But a company came up and infringed on the patent, and actually built a small company that lasted a few years. Maybe a little more years than your first one or second one, and but foolishly, their flagship, their mission was built around using a different CD34 monoclonal antibody to do the same thing, and so I spent a period of about 5 years, kind of stretched to really 7 years, in litigation and the post-litigation issues of this, and several lessons we could talk about later emerged from that. But I was subpoenaed, my assistant at that point comes in with somebody else, and this guy wants to talk to you, and he didn't want to talk to me. He wanted to subpoena me. And what do I do with this? And another thing was that you think that everything's clear, but I could tell you in small groups, the famous scientists who got up and said extraordinary things in front of a judge and jury, and this is professors as expert witnesses. He says, I know I'm perjuring myself, but I'm trying to make a point. You can't do that in court. You go to jail. So eventually we won. The good guys won. I had the experience of going to the U.S. Court of Appeals, and the bad guys, the little company, were found guilty of willfully infringing on the patent. And I love the judge saying, who figured it all out, that this was based only on greed, not on science or helping people. And they had to pay triple damages at the time. And it was a total waste of $40 million, which was a lot of money at the time, and could have been used for research on cancer or whatever you want. This is not the way to get your picture in Science magazine, or to get Harold Varmus to support your cause. But we liked the outcome, but I would have rather it been for an award or paper that my picture was in Science. But I do like this picture, and this was one of the first patients who got a CD34 transplant. And Brian, he had a soccer ball-sized advanced neuroblastoma in his belly and elsewhere. And we purified his CD34 cells from his marrow and gave them back after chemotherapy. And he and several other patients in the initial trial that I had the joy of doing from my own lab was responsible for some awards as well as therapy. But the real thing was that this helped people. And Dr. Seuss gives you all here in the audience the opportunity to help kids with cancer and their families. And as we saw from Will today, there's some extraordinary kids. Brian played soccer in high school instead of having a soccer ball-sized tumor. He graduated from the University of Maryland. He's an engineer now, and he has a family. And CD34 cells are still being used. It's still called the best antibody to isolate stem and progenitor cells, lo these 40 years. And it's a crucial part of the technique of hematopoietic gene therapy. These are the cells that are used that need to be concentrated and enriched before you can use CRISPR to repair, to either knock down BCL11A or repair the hemoglobin B gene with sickle mutations. And in fact, if you wanted to address T cell persistence, maybe you would put CARs into CD34 cells. That's been a fertile subject of research on another part of the planet. But anyway, so I've given you a little bit about doing research in childhood cancer or stem cell research. It can be really, different from what you thought. Neither my scientific or medical education prepared me any more than these guys are prepared. These guys at their microscopes, they're usually just looking in the microscope, and they're saying that there's more to life than they can see through the eyepiece of the microscope. And I won't illustrate this Gartner hype cycle, but you see the characters on this curve, some of them distinguished presidents and Nobel laureates, one an undistinguished president, and how that all plays on stem cell research and discoveries and patent laws and support for research. So I apologize because I've now spent the whole talk, and I'm out of time, talking about last-gen discoveries. I would have more naturally given you a talk about next-gen discoveries, so I'm obligated, I feel obligated to say, read this paper if you want to find out. One of the things we're working on now was a new chemotherapy regimen developed and dependent on a new drug to attack the MCL1 anti-apoptotic molecule in leukemias and other cancers. But this is a synergistic mechanism-based combination, which in models, including patient-derived xenograft models and primary leukemia-derived models, can make the leukemias disappear in the mice. So, the things that you will do, and please continue to use the Children's Cancer Foundation to leverage that, we'll call it $17,000 into hopefully 40 million of it won't go into a lawsuit, but instead will go into discovering, translating, curing, helping people, and along the way it helps to smile and to thank your supporters. So you'll have a lot of fun in this process, and don't forget your family in this process. They're your patients, but there's also your family. And again, thank you, CCF, and thank you all for listening today. It's been a pleasure.
Thank you, Dr. Civin. So I think that there's an emerging theme that we've kind of seen through these three talks, which is the ways in which CCF has really helped to launch careers and has brought forth really potent scientific ideas, all for the benefit of children with cancer. A few years back, CCF sort of recognized that this was a core part of their mission and codified that in a series of awards called the NextGen Awards, sponsored by Giant Food Corporation. And we have 4 of the last 5 years worth of NextGen awardees here. Taylor Sunby, we heard speak a little bit earlier, and Li Peng, who's also here from Johns Hopkins. Micah Maxwell is not here today. Haneen is another NextGen awardee, as is Jeff Rubens, who we've already heard speak today. So I think that this really is a really wonderful demonstration of the commitment to the CCF to foster the careers of scientists who want to help in this fight against pediatric cancer. It's not the easiest career. It's not the easiest way to get funding. It's in terms of NIH funding. It's a a difficult problem to tackle, but all of us feel the need very deeply within ourselves, and CCF really helps us to translate that in, hopefully into some new therapies and cures and improve the lives of children with cancer. So add my thanks to those of our panelists for the CCF and their support. I believe we have time for some questions. Okay.
I've got a question that was posted online, if I could ask. I think it's mostly directed towards Haneen. One of the worst, and this is appears to be anonymous, so I can't even say who it is, but please type in who you are if you'd like to be recognized. But one of the worst concerns with developing new therapies against cancer is recurrence of the cancer, either in the short term or long term. Are CAR T cells are the CAR T cells treatment mainly until remission, or are there maintenance regimens post-remission to lower the risk of recurrence?
That's a really interesting question, and I think one that we thought about for a long time at NIH, and actually just tried to kind of create a trial for maintenance-type CAR therapies. I think that they are mostly to attain remission. With CAR T cells at present, they are a living therapy, and so the hope is persistence. The hope is that the CAR T cells will continue to remain in that patient's body system. Of course, we are dealing with new things and complications that I don't think conceptualized at the onset of this, which I touched briefly on, which was antigen loss, antigen diminution. But, in fact, CARs do need some antigen in order to stimulate and expand. And so if you're giving them, as a maintenance type regimen, you need to be giving them in concert with something to promote that expansion. And there are clinical trials, that are utilizing tumor APCs in order to try to accomplish that. But I think that obviously, as we've heard today, research is super important. It is something that continues to propel the field, to drive the field. And I'll take this 1 second that I have to just say that as a NextGen awardee, it really was, it's overwhelming to finish all the years of training and then to get out into the real world and have people say, okay, now you have to go find ways to support your ideas and in what you want. And CCF, being a local organization, really helped kind of decrease that burden of anxiety for me as a young investigator. I can't speak for everybody, but it certainly helped with that idea that we had back then to do dual targeting. They grabbed hold of it, they embraced it, and with that grant, we were able to now, it's been 3 years, have 20 patients, treated to date. And so it's remarkable. And the community that it has inspired is really great. It was a fabulous idea to bring all institutions within this, small area together to be able to do this every year. It's very cool. I hope it gets to be bigger, as the years go on. But I know because I've seen the way that it's grown. And so it really is a pleasure to be here and to be with everybody, again, maskless and safe.
Great. There's another question from the virtual world, also CAR T related. Considering the recent developments in pre-tumor microenvironment research and genetic progression of cancers, do you believe CAR T cell treatments could be utilized as a preventative measure, like a vaccine, to attack developing tumors and cancer before they become overtly symptomatic? I know. I like this one.
That's a great question. There's probably more people in the audience that could answer that. I mean, the tumor microenvironment and utilization of CARs in solid tumors, I think, it is an area of very active investigation, and especially in the pediatric oncology branch, I know for certain that is something that we've been focusing on, and trying to target the microenvironment, but also trying to tackle changes in tumor cells and the heterogeneity of tumor cells in general. So the hope is, yes, that at some point we would be able to get that done. I think that there are a lot of researchers across the country and world that are trying to answer that very question. So I think more to come. Yeah,
one of the ideas that springs to mind every time I give a lecture on ALL is the discovery by Mel Greaves that the ETV6 RUNX1 fusion that is responsible for driving really the largest subset of childhood ALL. So in other words, it's driving one of the most common cancers in children. It initiates in utero, and unfortunately, it's not an antigen that's expressed on the surface of cells, because one could think about targeting that fusion in the 1% of the population that's born with that pre-leukemic lesion. Only 1 out of 1,000 of them actually go on to develop full-blown leukemia. So it would be really cool if you could identify the patients at risk with that pre-leukemic lesion and eliminate one of the most common, cancers in childhood. And I always tell the people I'm lecturing that your Nobel Prize is waiting for you if you can figure out how to target something that's in a pre-leukemic cell, so it's not actively cycling, and it's also not expressed on the surface of the cell. And I think that's, it brings up the challenges that CAR T cells rely on an antigen to be expressed in a tissue that's available, and that's not the case in many of these pre-cancerous situations that we're talking about. So I think that's one of the main challenges.
Is that antigen presented on MHC, the fusion peptide?
That's a great question. I don't think so, but I don't know that I've seen specific research on whether that fusion peptide is expressed in MHC. I think it would be really great if it was. That would open up tons of opportunity. I don't believe it is, but it's a great question.
I'll ask a question, and so we'll try and move off of Dr. Shalabi here, because you're ganging up.
That's what you get.
So, yeah. Give the most interesting talk, then you got to deal with the consequences. Yeah. So, Dr. Toretsky, so this, being somewhat of a retrospective, I was kind of wondering, you illustrated for us really a really elegant from idea to therapy, which is kind of unfortunately a somewhat rare progression, and it's also takes quite a long time to bring that to fruition. Technology advances, pretty rapidly as well. I wonder if you could look back at the journey that you took through the lens of current technology. Are there ways in which if you're doing it now, for the next person who has an idea that wants to take it forward, are there ways in which you think it would be accelerated from the way you had to do it?
So, yes, it can be accelerated, because a lot of the original work, say the first 5 to 7 years, were all in discovering these partner proteins. And a lot of the molecular biology that we used in 2001, 2002 has been superseded. There's technologies that will make that initial work, I wouldn't say obsolete, but would allow you to go faster through it. Just as one example is just sequencing. So when we did the original experiments to try to find the sequence of the DNA of these 36 bases that were going to lead to a peptide that ultimately led us to RNA helicase A sequencing reaction to get 36, actually about 50 bases, cost $25. Okay? So we went to our piggy bank at the time, and what CCF was going to provide us that, for that fund, and we figured we had a few $1,000s we could spend on that. It allowed us to sequence, 100 clones. Okay. Well, today with sequencing costs, you can do that for pennies. I mean, it's absolutely pennies on the dollar, and you can sequence 1,000 clones. So that's the first thing. Having said that, then the other steps of the way, we're learning more and more about, how the proteins themselves interact. And that is such a hot topic now that there have been literally a number of companies that have formed in the last 3 to 5 years with experts who study these protein-protein interactions based on what would be called either intrinsically disordered proteins or multivalent interactions that lead to this new phenomena called biomolecular condensates. Okay, so this is a huge area of research both for cancer and neurodegenerative disease, trying to find drugs that will go after these areas, and that technology is also advanced. And so between all of that, I think that, there's a lot of reasons to think things could go faster. On the other hand, there's some things that you can't speed up. In one of our early trials, there was a safety trial that the FDA required us to do and set us back 6 months because the company got in animals that when they put in the catheters to the animals, unbeknownst to the company and us, there were bacterial balls at the end of each catheter. So as soon as the animals were injected, they all died of sepsis within minutes to hours. And it turns out that the company was ready to even drop the project at that time. Except I said, do a postmortem on these animals, and they were all just loaded with bacteria. But the problem was now you're at the back of the queue for the company who needs to do that study for you, and you've got to wait 6 to 9 months just to get to the front of the line again, even though it was their mistake. So there's some things, yes, that will speed up, and there's some things that won't.
How are we on time?
Good. We've got 14 minutes and 5 seconds left. So if anyone has questions, either virtually or here in person.
It's a general question. With the new administration in D.C., there's a lot of federal dollars that are going out. Are you seeing any opportunities for any more or any better chances for funding than today than, say, 10 years ago, 5 years ago?
Well, the newspaper yesterday says that the NSF, or that the president will propose legislation so that the NSF gets a big boost to boost our science in the competition and in the post-pandemic realization of the things, the advantage of being in an advanced research environment to that. We don't know what will happen with the legislation. Will we have an ARRA-like period that we experienced in 2010 or so after that recession? We certainly hope we will. And remember, President Biden, as vice president, was the symbol of the moonshot and really worked hard on that. And so I have to believe that he will fight through support for cancer research. And as part of the moonshot, pediatric cancer had a position in that. So that my answer would be fingers crossed, but my answer is based on reading the newspaper and talking to folks just like yours.
One thing I can add to that is a really nice development this year is the Department of Defense has traditionally done a lot of cancer research funding, but it has disproportionately gone towards adult cancers. And there was a group led in part by the St. Baldrick's Foundation, some other foundations, to really lobby for a larger proportion of the DoD budget that goes into research to be towards childhood cancer. And so for this year, they've earmarked something like $60 million, $65 million, which is a significant proportion of the budget specifically for childhood cancers. And there's a big push right now to make the pediatric cancer research community aware that this funding is available. And so that's something that, would be, it really is a good sign. I think that from the federal government perspective, at least that slice of it, there's an increasing recognition of the need for funding for childhood cancer research specifically.
But this will not make, whatever funding is, will not make the need for foundations like the Children's Cancer Foundation go away, because always there will be 10 or 100-fold applicants for every award. And I review a lot of those awards, and you're never going to award something at the crazy idea stage that
That's right.
several of us talked about.
It's really the preliminary data stage that the foundational support is so, and you've got 3 people sitting up there that have just demonstrated that so clearly how important that is at an early stage.
Even if you want to do that, if you've got 10 other awards in the pile that had a crazy idea and now it's feasible, shown to be feasible, you're going to fund those.
Yeah. I mean, the current, what they call pay line, which is at what % does a grant get funded, over the last 3 to 4 years for the National Cancer Institute, and this is for all grants, not even childhood cancer, has been sitting around 8%. So in other words, 8 out of every 100 grants are going to get funded, which means 92 aren't. Now, of the 92, one might throw 40 or 50 of those off the top because maybe they need more work, maybe they need a little more polish or something, but there's a good 10 to 20 grants in that stack that are getting randomly discarded because there just isn't going to be the funding. Now, as the pay line starts to move up, which is one of the points that some of this additional funding might might allow. Maybe you'll get to 12 or 15%, but that's still, you're still going to be discarding a number of grants that are going to fall just outside that probably could be also really good ideas. And so it's really been a phenomenal thing that CCF can provide that kind of data that might take a grant that was in the 20th or 22nd percentile, still a good idea, and move it down to where it could potentially get funded. And in fact, that's what happened to our first grant. So when we tried to get the first grant to move this drug forward through the NCI, I had to submit that R01 grant 6 times. Okay, took 6 times. Finally, on the very last time, with all of the data that we had, the thing scored 0.8%. And I originally called the program officer.
That
Was
means
there
top 1%.
Was there a decimal error there? Did they get the decimal point wrong? And they said, no, yours was finally, after all these submissions, you finally got the top spot in the pile. But that's what it took. It took the persistence of sending the same thing with improving it each time with the preliminary data that CCF was allowing me to keep the project going to get to that 6th submission. And I hope others don't have to get to that point. But if you do, that persistence can sometimes pay off.
Kudos to you for having that persistence. That's
There's a word in Yiddish. It's called meshugana.
Means crazy.
Another
Gesundheit.
development that's not strictly funding, but I think is equally important, is the RACE Act, which was passed about a year ago. And what the RACE Act does, for the audience who doesn't know, is when there is a genetic mutation or other genetic anomaly that's present in an adult cancer for which a pharmaceutical company is pursuing a therapy, and if that abnormality is also present in a pediatric cancer, then they are now obligated to also investigate the pediatric indication for that, which is really hugely important because for so many years, the pediatric market is really not going to incentivize pharmaceutical companies to really go after pediatric specific targets. And so this really kind of, I think, helps us be less of the beggars in the room, and hopefully they're going to need us now, and we can hopefully marshal some of those pharmaceutical resources as well. That was not possible in the past.
That's great. Any other questions? Let me just check the virtual question box, see if there's any
While you're
more.
checking, Pat, another point on that about that's valid at this point in time is we all know that cancer research has slowed down. I mean, we've managed to sustain it, but it's slowed down during the past year and a half of pandemic restrictions, and cancer hasn't slowed down, and so we need to catch up.
I also want to recognize Dr. Brown and I guess conclude your morning moderating efforts. So thank you very much for taking on that role. And we'll welcome Dr. Dabe in the afternoon, who will take over. So we'll hand the baton to you this afternoon. So unless there's any other questions or needs, we'll take a break for lunch. And thank you, everyone.
Thank you.
Thank you.
That was fun.